Okay, cool. So welcome to Lab 6. It's kind of a fun lab about overgrowth and succession and competition and all this fun stuff that we've been building up to. So it should be pretty fun. It's unfortunately definitely not the same as in normal times when in the lab you get to actually handle these plates, but hopefully getting to look at some of these pictures of these creatures is fun as well. So before we move on to talking about the lab, does anybody have any questions about last week's lab or this week's pre-lab? You can type them or unmute yourself. Give you a second. Okay, not seeing anything right now, but just in case, I think I sent out a message to this effect, but I recorded my kind of whiteboard session going through the math for last week's, at the end of the section, I went through all the math that you'd be doing for the post lab in the recording, so that should be on our section's YouTube channel. And you can review that if you're having trouble with any of the equations for the post lab. And remember, the post lab is due tomorrow by the end of the day. Then you have the next pre-lab due on Monday, and then you also have a midterm on Monday. So on the 17th, I think. So just a few dates to keep in mind. In terms of the pre-lab questions, people did really well on this pre-lab. So I didn't have any specific questions that I felt like we needed to review. Maybe just the one that it was asking what indication. Let me see if I can pull it up. What indication that there was a shortage of resources or space, and I can't find the exact question, but the answer there was overgrowth, and that overgrowth was indicating that there was a shortage of space, that you could see that these creatures were actually growing over each other. So maybe when we have some downtime, I can check on that exact question. All right, so moving on. So the topic of this week's lab and post-lab assignment are succession in a marine fouling community in Bodega Bay, California. Has anybody been to Bodega Bay ever? No, it's so beautiful. I really recommend it if you have the opportunity, if you have a car, if you have a friend with a car. I really recommend going out there. Davis has this marine science center, and a lot of the people in the ecology program who do marine stuff, they are based out of Bodega Bay Marine Lab, and it's just a really cool facility. Sometimes they have classes that visit there. They have opportunities for undergrads for doing research there. So if I see anything, I have a few friends who are out there, I will definitely send it to the class. But if you see anything pop up, highly recommend it. Such an awesome place. But they also work with the biz classes in terms of kind of providing different experiments that they're doing for us to look at. And then one of them is these plates that they've put out, and we can see the kind of communities that are growing there. So it's super cool. And yeah, it's like about maybe like a 2-hour drive from here, but just north of Marin in San Francisco. Yeah, great place. So we're talking about succession, and you may have, this is a really commonly used graphic when talking about succession. And a lot of times when succession is introduced, they talk about kind of like a forest community, I think they often use, but it can happen in all different kinds of ecosystems. So we're looking at this tiny, tiny scale marine ecosystem, but it also happens on huge scales. And you might have thought about it in after disturbance or after fire. So can anybody tell me what succession is, how you would define succession? Not like that HBO show, but I guess kind of like that. I haven't seen that show, but. Not seeing anything yet. You can also send it privately to me if you don't want everyone to see. All right, so yeah, evolving over time, great. So yeah, no, I'm seeing evolving over time. Yeah, evolving over time is kind of a good way to think about it. It's how, rather than like individual organisms, we're thinking about how communities are changing over time. And so you can think about maybe after like some huge disturbance, like you could think about maybe a volcano and everything gets covered in magma and everything that was there is now covered in rock. And so what kinds of plants and creatures are the first to like start to inhabit that new substrate, as we call it? And then kind of how does that community change over time? Because probably, as you can see in this graphic, you know, the first thing, there's exposed rocks, and then maybe some little lichen guys and mosses start showing up. So it doesn't stay like that necessarily for the next 1,000 years. Like over time, maybe grasses start coming in, then some smaller herbaceous plants, then shrubs, a young forest, mature forest, and then what's called the climax forest. So we will see this change as, you know, as soil develops, maybe taller plants are able to establish themselves, maybe on a new substrate that seeds haven't been able to get there, so there aren't certain plants. So as we're thinking about succession, we're thinking about how these changes occur over time and then why they occur over time. And then as we're, you know, biologists, we love to try and understand the system. And so we're thinking about how we might quantify a change in community structure through time. So this kind of ties in with a lot of what we've been talking about with sampling and how do we, you know, quantifying biodiversity, for example. That could be one way to look at change in community structure. We could look at percent change cover if you're just looking at it. That's kind of what we're talking about today. And then we're also thinking about, after we're thinking about how to measure these changes, we're thinking about what mechanisms can explain why there are changes in the community structure. So, you know, how to develop a hypothesis to understand these changes, and then really how do we test our hypothesis. So this lab is kind of the culmination of a lot of different stuff we've talked about over the quarter. We're talking about biodiversity and diversity indices, and we're talking about sampling and estimating cover and all these things. And then we're also talking about developing a hypothesis and figuring out how to test it. So it'll be a little bit of a combination of some of the labs that we've done before. And in the end, we can talk about developing a hypothesis similar to how we did a few labs ago, and really figuring out if you could go out in the world and do an experiment, what you would want to do to see how these communities are changing. Any questions about the topic of succession or what we've talked about so far? All right, so these are all links that I believe are also in the lab manual, but there's an intro video to this that's kind of cool, and it takes you actually out to the Bodega Marine Lab area and where they collected these plates from that we're getting our estimates from. And then there's the data collection tutorial, so how to get the data for this lab. And then, so for the lab, if you got the kit, you have these funny images, which are pretending, you know, pretend these are the actual plates and they have the different creatures on there. But if you didn't get the lab kit, or the color is not super bright, So even if you're having trouble identifying the different species on here, you can use these data collection images, which is on this Google site with these fouling plates, as they're called. And you can use that for your estimates as well. If you are using the paper sheets that you got in your lab kit, it also came with this clear plastic sheet, which you can use to get the estimates. And that we'll go through that a little bit, and then it also goes through it in the lab manual, how to use this to get the data. So as we talked about a little bit earlier, going through succession, our basic premise is that community structure changes through time on different substrates, and that can be as huge as an entire island emerging from the sea after a volcano erupts, like i.e. the Hawaiian Islands, or it could be as small as a tiny piece of PVC tile that is placed in off the Bodega Marine Lab docks. And so when there's this new substrate, this new, area for species to start developing on, there are the first ones that come are these things called colonizers or pioneer species. And oftentimes we think of these as R-selected species. So like we've talked about in lab and in class, we talk about R-selected species and K-selected species. R-selected species are going to be their population growth is only limited by their reproductive capacity. So they can just explode in growth, and it's basically their population growth is controlled by how much they can reproduce. And they're often like quick reproducers, and they can reproduce like a ton at a time. So we've talked a bit about that, and that's frequently what the colonizers are. Maybe think about like a dandelion it has all those seeds that explode off of the head and they just go everywhere. And so say they just blow on the wind, a million of them blow on the wind, and they end up on this new substrate where there's nothing else growing, then they're just gonna establish really quickly and kind of colonize that new substrate. Then there's this mid-successional species comes next. So this can be kind of a mix of R and K selected species depending on what areas are available, how much space there is, what gets introduced. So you're kind of starting to see more of that K selected, which is more of the slower growing, maybe limited by carrying capacity more than growth. And then later, we call the late successional species also a climax community, and this is going to be mostly K selected species. So this is the general concept for understanding how succession occurs. There's lots of nuance in it, and this isn't actually, totally the whole picture, and there's always exceptions. But in general, just kind of think of this progression of like these colonizers or pioneer species come in, they're super fast reproducing, and they just have a bunch of, babies all over the place to take over the substrate. Then this mid-successional species, which is a mix, some quick-growing individuals, some larger, slower growing individuals, and then the later successional species, which are mostly, slow growing, K-selected species. In the forest, you could think of like trees as these long growing, long living, giant successional climax community members. All right, so our system, like we said, there's the forests and the islands, and we have these tiny organisms growing on these little plates on a floating dock in Bodega Bay. Kind of looks like a modern art project or something when they're all together on here. These actually really pretty colors, I think, like mustards and purples and stuff. So it's fun, be even more fun if we got to see them in person, but we will be happy seeing pictures of them as well. And so we have the difference here, the different treatments, if you will, in our experiment is time. So you have a plate that has been in the ocean for 3 months, then a plate that has been in the ocean for 6 months, and a plate that has been in the ocean for 9 months. And we're kind of seeing how does the community structure change? This is a microcosm of that succession that we've been talking about. So how is the same plate or even groups of plates changing over time as a whole, the longer that they've been in the ocean and the more they're exposed to all these different species that can come and colonize them? So there's going to be two parts to this lab. And the first part is our data collection. So each student will sample 3 plates. You have a 3-month plate, a 6-month plate, and a 9-month plate. And the data that you're going to collect is the number of species. There's an animal guide on Learning Tree that you can use to identify the species. You don't have to identify it to, scientific name. I think if you want to and you can feel like you can identify them based on the guide, that's fine. If not, you can just make sure you're being consistent, like, oh, the squishy looking yellow thing and the purple polka dot thing, something like that. Kind of like how we did in lab 1. And then you're going to get the total percent cover for each species. So how much of the plate total is covered by each species. And then you're going to get percent cover on the panel surface only. So only something that is actually touching the plate itself. And we'll talk about that a little more because I think that's kind of confusing to get the difference between those two. But basically you're trying to get amount of overgrowth and how much, species are overlapping on this very limited area that they're growing on. Sorry. So once you've collected that data, then you're going to be doing some calculations that are mostly familiar. You're going to be doing a diversity index like we did in lab 1. You're going to be looking at that percent overgrowth. Then you're going to pool data with previously collected data. So you are just getting this one sample, and there is going to be a spreadsheet on Learning Tree. I believe that it's on here. Yeah. There's lab 6 data, and it says add your data to this data sheet. So the data sheet has all these other samples already collected, and you're just going to add your 3 samples that you get from those 3 plates to the data sheet. And you're going to make graphs with that data, getting the average values for number of species, total percent cover, diversity, percent cover on the panel surface, and then percent overgrowth. And the lab manual will also walk you through those and the post-lab instructions for which graphs to make. Okay, so going over how to do the data collection, you have this picture of the plate. So either on the website, like we talked about, or on your sheet that you got in your lab kit, and you're going to place this grid over it. And then you're going to get estimates based on the number of squares or the percentage of the square that it covers for how much is covered by each species. So the reason that we're not doing number of individuals like we did in lab 1 is because some of these are colony species, or they're really hard to tell which an individual, or they are all growing in some big mass, or there's a million of them on one plate. So rather than make you count like 7,000 different little tunicate guys, we're just going to do percent cover. So hopefully that's easier. But let me get the instructions up. So let's say what percent each square is. All right, so each square is 4%. Yeah, so each square is 4% of the entire plate. So when you're doing this, like say, let's find one that's 100%. So like this guy right here, we could say he's almost 100% covered with this tunicate. So this is 4%, this guy's mostly, this is another 4%, another 4%, another 4%. So you count up how many percent total are covered by this one species, and then you would put like, okay, so there were, let's say 60% of the plate is covered by species 1, which is this tunicate, these kind of tube creatures that are super weird, and you learn more about them in BIS2C. And they're actually like not that distantly related to us. So they're crazy. So then you collect for that one species, and then you look at this kind of yellow and black type thing. And even if it's growing on the other species, for our first measurement, we're still going to count that percent cover. So we're like, okay, maybe it's 1% because it's like a quarter of this square, and maybe plus 2% because it's half of this square, plus 3% because it's 3/4 of the square. So adding all that up, and then you'll get the total cover of everything that you can see, or even if it's under it, if you still think it's there, like if this was growing under the tunicate, but you still think it's there, you'd still add that to your count. So you're going to go through all of that and collect for each species total cover that you think is going on. Let's see if this, I forget my slides. Yes. So total cover that you think is there. And then you'll do the diversity index calculations. So like getting this P1 or PI value is out of the total cover. So on this one, I'd say not 100% of the panel is covered. So out of all the total cover that you think there is, what percent is covered by that species? So they said only 80% of this panel was covered. So if you said 20% was the crusty thing, then the PI value for the crusty thing is going to be 20 divided by 80. So a quarter. Excuse me. You'll do that for all the different species that you have. Then after you've gotten this PI value that you can use for the diversity index below, you're going to get the percent covered that's only on the panel surface. So the percent covered that is directly touching that PVC panel. And I know it's hard with, it's a picture, so just do the best that you can. But for example, here, this like yellow thing that's growing on the other guy, we would not count that for percent cover on panel surface because he's growing on another creature. So do your best to identify what's growing directly on the panel and what's not. So they say the crusty thing, I don't know, this is from Pat, but I think that the funny thing maybe they mean is the tunicate. So say the tunicate is the funny thing, and all the tunicates are growing directly on the panel surface. So if you have the percent cover on panel surface, all of the things that are growing on the panel are growing on the panel surface, it's just going to be the same number. So 30, 30. Some of the things, say the crusty thing is the orange guy that's growing on top of other things, you're just going to count what percentage of the orange thing is growing directly on the panel surface. Then once you get that estimate from what's on the panel surface, you're going to get this percent overgrowth, which is just going to be this column A minus column B. And that'll give you the overgrowth. So what percentage of what you measured is growing on top of something else? Any questions? I think it probably sounds more confusing than it will be when you're looking at it. We can also go through it at the end again. Okay, so just like we did in lab 1, you'll get this, you'll look at the number of species, and you'll be able to use these PI values to do a diversity index, and you'll get percent cover on panel surface and percent overgrowth measurements. So the data sheet that you're going to download in the learning tree manual, let's see if I can just get it up. So you can see it. Yeah, so this is what you'll download from Learning Tree and see it has all of this stuff already calculated. So it'll be pretty easy for you to just, here's your data, put in the number of species, total cover, the diversity index value that you calculate, the percent cover on the panel surface, and then percent cover as overgrowth. And you have that for 3 months, 6 months, and 9 months. So this is the overall data. I think this, the way it's on the slide here is a way for, this is more like what's in the lab manual. They have these kind of data sheets, data collection sheets for you to do. And then once you've done those data collection sheets, I can show that to you. So this is what's in the lab manual. It has this data collection. This is basically just what's on the slide, and you'll do by species, percent cover, the PI value. So that's the proportion of it that is that species, percent cover on this panel surface, and then the overgrowth. And then once you have filled out that, then you can go over to this handy dandy data sheet that is on Learning Tree and put your data in for 3 months, 6 months, and 9 months. And then you can make the graphs that you need for this assignment. Oh yes, more Excel. I know, welcome to science. So much data and Excel. Wait, how did you get that emoji in the chat? Anyway, you can tell me later. I'm very impressed. Okay, so that was lab 6 part 1. For lab 6 part 2, we're also going to be proposing a hypothesis like we did a few labs ago. So basically for this part, you're going to pick one pattern that you noticed, that you see as you're looking at the data. So for example, an example here is overgrowth. Oh, wow. Okay, so I have an answer to this. Keyboard shortcut for emojis. Okay, well, I have a Windows, so sorry for those watching at home. This is important keyboard shortcuts for typing emojis into chat, very important. Okay, so back to hypothesis. So you pick a pattern. So say overgrowth is greatest at 9 months. So you're looking at your data and you see that, okay, so many of the species have the highest percent overgrowth in the 9-month tab. And so you're going to propose a hypothesis to explain the results. So say, I believe that more overgrowth is a result of lack of space. There's all these species trying to colonize, and these plates have already been colonized, so they're just growing on other critters. And then you're going to propose methods to test. So say a method to test this could be that you place different size plates, really large plates, medium-sized plates, and small plates, and then you see if the percent overgrowth on each plate at 9 months is different. So if you believed, if your hypothesis were true, if you thought that more overgrowth is a result of lack of space, what you expect to see is that on the large plates, there would be less overgrowth than on the small plates. I've just heard word that there is a keyboard shortcut for Windows for emojis. This is exciting. We'll do this when I stop. We can play with this when I stop recording. So this is one idea. Other ideas for your hypothesis could be competition, predation, environmental influences, mutualism. So you could say that you think that growth would be greater in areas with overgrowth because a mutualism is happening, and you could test this kind of measuring the same individuals and seeing how they change over time, for example. There's lots of different ways to go with this. it's a similar thing where if you had unlimited time and resources, what kind of experiment would you want to design to understand a pattern that's going on here? So we can talk a little bit more about your hypotheses. After this, I'm going to do a quick run through of the organisms. And then if you're watching at home, you can also come to my office hours if you want to kind of, just like we did in the other lab with hypothesis testing, go over your ideas and make sure that it's making sense. You can come to office hours on Tuesday or Thursday morning. But for now, we're just going to go through quickly some of the organisms that you will be seeing in the pictures. And like I said, you don't have to call them by their scientific names, but it can be fun to know what they are. So these yellow guys, kind of, oh my gosh, they all look like aliens to me. But these kind of orangey, like mac and cheese colored guys are tunicates. And so each whole like little darker circle that you see on there is an individual. And because we wouldn't want to make you count like a bajillion of them, this is, you know, why we're doing percent cover. These kind of grayish and reddish guys over here are the bryozoans, and they're also like each one is a little individual tube. These are mostly modular creatures, so you've been introduced to those modular unitary terms. So these guys are mostly modular. Then we have our unitary animals, these marine worms. We have other like those big tunicates like we saw in the picture before. And then you could have this spaghetti worm with a mud tube and foraging tentacles. Oh my gosh, they're so crazy. And if you're taking BIS2C next year, and hopefully it'll all be in person, you have a super fun series of labs where you get to like actually handle a lot of these creatures and see them up close. And there's tanks in what is usually the BIS2B labs, but there's a whole series where you get to really get up close and personal with some of these creatures. So very exciting for all of you who are going to be taking the class in person next year. And whoop, stop share. So yeah, like I said, there's also the animal guide on Learning Tree for you to go over those if you want to use that for identifying the animals. But if not, you can call them whatever. You can call them Fred if you want, but maybe put something identifying in there so I know which creature you're talking about. Are there any questions about this stuff? Watch us just call all the creatures Fred. Yes, you could, as long as you're differentiating them, you could be like orange Fred, yellow Fred, black Fred, red Fred, I guess. So any questions on this so far? At least the chat has been lively. No questions here. Awesome. That's what we love. Okay, so I'm going to stop the recording for now, and we can also go through some of the sampling scheme and using the spreadsheet if you need, or other questions or hypothesis testing. And for those at home, good luck. Come to office hours if you have other questions, and hope you all have a wonderful week.